Hello, my name is Arnaud Delorme, and I'm the main software architect of the EEGLAB software for processing brainwave data. So in this series of video, I'm going to show you how to pre-process data in EEGLAB. And the first step is to import your data into EEGLAB. So first, why pre-process data? Well, EEG data out of the recording device is a continuous and process signal. So it's like measuring a difference of potential on an oscilloscope. And you can actually do that if you want. You can measure brainwave data on the oscilloscope if you wanted to. So to make sense of the data, we will need to extract meaningful measures from it, such as brain oscillations, brain source activations, compare brain data in different conditions, or assess reliable changes to external stimuli, event-related potentials, for example. But before we do all that, we need to apply a series of transformations to the data. And these are listed here. This is the standard pipeline for processing EEG data, whether you use EEGLAB or any other software. First, you would collect the data, then you would import the data into EEGLAB, then import event markers and channel locations, which are very often in separate files. Then you might want to re-reference your EEG data, or also decrease the sampling rate of your EEG data. Then you might want to filter the data, which is here. At this point, it's good to actually look at your data to see what kind of data you have and what kind of artifacts are present in your data. Then you would want to identify, reject bad channels. Very often when you collect EEG data, some of the channels are bad, they have a bad connection. And then reject large artifacts, which are very often present in EEG data. Every time you move, for instance, there'll be like a dramatic artifact that needs to be removed. And then the last step in this pipeline is to run an algorithm that's called independent component analysis, and that's basically an algorithm that allows you to extract a stereotyped artifact. It's very powerful and widely used in the field, and then you have to select components to remove the components you want to exclude from the data. So the first step is to download and install Matlab, and download and install EEGLAB. So Matlab, you have to pay. That's a commercial software. EEGLAB is free. You don't have to pay for EEGLAB. You can run a lot of the EEGLAB under the free Octave software, but you don't have any graphic or interface. You can plot most of the graphics, but you can't use the graphic or interface of EEGLAB. So download EEGLAB. It's just a zip file. So you unzip the file, then you start Matlab, and then you add EEGLAB to your path. So either from the command line here by typing something like this line, it depends where you unzip the EEGLAB, of course, or you can set the path here. And if you set the path, there is an option to add all the subpath. Don't add all the subpath, just add the main EEGLAB path, and EEGLAB will take care of adding the path it needs to add if that becomes necessary. So then you type EEGLAB, and some of you might have the compiled version of EEGLAB, which doesn't require Matlab, so you don't have to do all these steps. You'll just get the graphic interface directly, like this one. So you type EEGLAB, you get this graphic interface, and then you're ready to import data. So there's different options to import data. First, so that's the first menu, File, Import Data, and you can select native EEGLAB functions and plugins. So they're listed here, and here you should see the format you're interested in. So it supports most of the main manufacturers, and then for all the other ones, there's like dedicated plugins. We also interface two other toolboxes. So here it's called the File I/O interface and the Biosig interface. And these are separate toolbox for EEG under Matlab, and these allow you to import mostly the same formats, but also different formats sometimes. If your format is not listed, what you can do is you can go to Manage EEGLAB Extension, Data Import Extension, and then you can see in the Import data extension, if your format is listed, then you would just check install, press OK. It would restart EEGLAB, and now there would be a new menu in the import data from using EEGLAB menu, where your format will now be listed, and then you can use that additional menu. These extensions are developed by third party. You can develop an extension if you want to. So that's why they're not in the default EEGLAB interface. And if you go on the EEGLAB wiki, there's also a list of file formats which are supported, and they're listed here. And there's different columns, either it's supported directly by EEGLAB, by BioSig, or by File.io. You can also go to the BioSig website, which tells you all the formats which are supported, or the File.io websites also contain some information. So I'm going to show you how it looks like. I've just started MATLAB. If you haven't added EEGLAB to your path, you can do it here, set path. And here, add folder. I'm already in the EEGLAB folder here, so I'm just going to press open. And don't add with subfolder, although if you do, that's not the end of the world. I would save, close, and now I'm ready to start EEGLAB. This shows you the plugin you have installed at startup, by the way. I have more plugins here because I've installed these plugins. So here is the menu import data, which I mentioned. And I'm not going to use this menu right now. Instead, I'm going to use this menu, load datasets. That's loading EEGLAB dataset that has been saved. So after you import your data, you can save the data as a EEGLAB format. And that's the data has been saved just after importing the raw data. So here I'm going to the EEGLAB folder, and then there's a subfolder that's called sample data. And this EEGdata.set is an EEGLAB dataset that has been saved just after importing some raw data. And as soon as we import, we have some information here, like the file name, number of channels, number of samples, epoch. That's when it's continuous data, it's always 154 events, 128 sampling rate, beginning and end of the data in seconds. The reference, it doesn't have channel location yet, doesn't have independent component analysis calculated yet, and the dataset is 4.3 megabytes. As soon as you import the data, you might want to look what's in the data. Is there any data? So that's the menu plot channel data. Plot channel data. And here you can see your raw data. So here I have 32 channels. If you don't see your data in that plot, don't panic. We'll see how we can filter the data to bring it back in range. So that's it for importing your raw data into EEGLAB. And in the next video, we'll see how to import events and channel locations.